and it's going to be 2 layers of them that's going to be inside, and inside of that, there could be a ton of proteins and other stuff. There could be glycolipids, there could be glycoproteins, there could be transmembrane proteins, all different types of enzymes, you know, everything that's embedded. It's not just a phospholipid bilayer, there's tons of stuff in it, and there's all kinds of rules for how it works. So this is one of the first videos that I was talking about that you would definitely want to watch, and this just gives you a general information all about the cell membrane, and so they go piece by piece and all the rules for it and everything else. If we get to it, that's great, but again, we might not today. So for the lipid bilayer, it's actually hard for me to see his turn, but what are the components again of phospholipids? And just remember, you're going to end up having a fatty acid chain that's going to hang down. You're going to end up going and having a phosphate group of some kind and a 3-carbon alcohol. And so again, this is the 3-carbon alcohol. It's usually like glycerol or something like that. You'll have a phosphate group. This can vary depending upon the type of phospholipid, so you can ignore that part. But a phosphate group, usually glycerol, like a 3-carbon, 1, 2, 3-carbon alcohol that we have. And then there's the 2 fatty acid chains. And so that's what's going to be located on there. That have to have those 3 parts. So the part at the top just tells the different ones, but you have to have at least those 3 main parts, otherwise it cannot be considered a phospholipid bilayer. And so, again, yeah, it's going to have a phosphate group. It's going to have a 3 alcohol, like 3-carbon type of molecule, and so usually glycerol or something like that. And then it has to have 2 fatty acid chains. And so that's what they're trying to show you here is it's called a bilayer because you have one here opposite each other. The polar hydrophilic heads will interact well with water, and then the nonpolar are going to shrink away from the water, which means water cannot get through here very easily, so this way it's going to be able to protect it. Again, there's going to be aquaporins and other stuff that goes through. And so it's going to protect it from the outside for the most part and make sure like nothing is going to affect it. So again, that's what it means by a bilayer. You have one facing like this, one facing upside down. The tails will touch each other and the heads are going to be opposite each other. And so this is how the main setup of your bilayer is, but inside of it could be like transmembrane proteins and other stuff. So what they say is the lipid bilayer is fluid. So it's not that it's like filled with fluids, it just means simply that it's not going to be like very static. You can actually have the membrane, have the proteins, like the transmembrane proteins move a little bit. They're not always just going to stay in exactly one place. I mean, unless the cytoskeleton really anchors them in there so they can't move at all. But for the most part, we say that it's a mosaic of proteins and that they're going to be basically float in or on the fluid bilayer like a boat on a pond. So that's what it pretty much will remind you of if you actually saw these things under like an electron microscope. And so the proteins that are inside your lipid bilayer don't always just stay still. So they can move around, they can stay on the actual bilayer, but it's almost like boats on a pond floating that you will sometimes, encounter. And so that's what they're trying to show you in that picture is if you go and you take the lipid bilayer of a mouse cell and a human cell, and they're going to contain the proteins on them. If you actually fuse the 2 cells, again, this is like on purpose. We're not creating mouse humans. I'm just saying like the cells themselves, the membrane, if you do a comparison, the actual proteins, they intermix with each other when you do this. And so in other words, what ends up happening is they're not going to just be static. They're not going to stay, half of it, the globe will say is going to be red, half is blue. They do mix. And so at least some membrane proteins can diffuse laterally within the membrane and go all over the place. That's what I mean by boats on a pond. It doesn't just stay in one location. And so that's why we say the lipid bilayer is fluid. It's simply that you're not going to have all of the membranes just be bound and stuck there in the cytoplasm by the cytoskeleton and the extracellular matrix or anything like that. They can move. And so again, the main thing and the reasoning this is the case is hydrogen bonding. That's what's going to be going and holding together a lot of this and hold the membrane to make sure that it's going to, again, be continuous bonds, breaking and forming. Because remember, when you have something at a certain temperature for water, it's 10 to, I think the -7 seconds of how fast the bonds break and form. And so because it's doing this so quickly, that's what allows fluidity. And so that's why water is fluid. So it's all due to hydrogen bonds breaking and forming continuously, and that's why ice, they don't do that. And gas, you don't even have hydrogen bonds because they're just floating around, the water molecules. Van der Waals forces are the other reason they stick together, because as long as everything is packed tight, Van der Waals forces will be enacted. Remember, they're very weak bonds. They're the weakest bonds you can have. Both of these are weak bonds. But the point being is if you have high polarity, hydrogen bonds can definitely be forming. And then with Van der Waals, they act like Velcro. So if it's really, really close, you're not pulling those things apart. If it's farther away, is spaced differently, then they are going to more than likely, disintegrate. But those two interact with each other, those types of bonds, allow this fluidity to happen. So unanchored proteins, in other words, can move within this membrane, and again, that's due to the weaker bonds. The fact that these two are not polar bonds, these are going to be hydrogen and Van der Waals. And so the unanchored proteins can actually move around. And again, the cell fusion thing I put that's with the example, but I already had gone over it. So the other reason you do need cholesterol, I know some people think like, oh, it's a bad word. It's actually not. You have different types of cholesterol in your body. You have LDLs and HDLs. LDLs are bad, HDLs are good. The HDLs, you actually want a certain amount of them in your membrane. And so cholesterol is technically a steroid lipid. So what it does is it can either increase or decrease the membrane fluidity. It just depends on the temperature and how much is going to be there. But you do want it to be present because it actually helps with the rigidity of your actual cell membrane. And so again, the way the steroids of the cholesterol work is they can either increase or they decrease the membrane fluidity, but it's all based on temperature. And so if you're going to have a lower temperature, things won't move as much. If you increase the temperature, things will definitely move, but the problem is there's going to be a limit, because just remember, not everything can survive forever. If you go and put your hand in boiling water, the entire thing's going to burn, it's going to hurt like anything, and your hand's going to have a lot of dead skin cells on it. In other words, high temps are not good. And so that's why when you get a fever, things can happen in your body. Even your brain starts to basically melt at 105 degrees. And so, internally. So not good. And so that's why you can get brain damage, and that's why they tell you if you have a really high temp, go to the freaking hospital. So when it comes to cholesterol, again, that kind of helps all of that. But if the temperature is too hot, it's not going to help. So it's just going to end up going in the equivalent of almost melting, and it's not going to do very good. Can't repair itself, and you're going to have some problems with your cells.
That's just right. Is this limited to only red blood cells, or is it also like
all cells?
Present in all?
So every single cell will have the same general buildup. It's just that the different types of proteins can vary, but they all have some form of cholesterol. So in certain locations. Yep.
Can you repeat unanchored proteins?
Simply that a lot of the proteins are not necessarily connected to the cytoskeleton, so they can move around. But again, it kind of has to do with the temperature, and it has to do with the way the bonds work. So because of the hydrogen and Van der Waals, they're not strong bonds. So what can end up happening is it allows unanchored proteins to move. And that cell fusion is just an example of that. And so that can happen if they're not going to just be attached down and not moving in any capacity. So that's why you can end up having them float all the different around. Now, is this like a viable thing that could be alive? No, we can't have like, mouse humans. But it's just the point that the membrane was all they were studying, and it can be intermixed. Not the DNA stuff, that's separate. This is a cheat sheet for you of just, in general, the different components of the cell wall membrane we're about to discuss, all the functions of them. Again, we're going over more examples, so this is not going to be as detailed as what I'm telling you, but if you get stuck on something, this is the place to go for studying. And again, it gives you some examples. Are we going over all these examples? No. But it just gives you at least an idea of what some of the different types of cell membrane parts they're going to be. So, the fluid mosaic model, this came out, I believe, in like 77, that they agreed upon, like this is what the membrane looks like. Oh, 72, sorry, with Singer and Nicholson. So what they basically said is globular proteins. Whenever you see the word globular, it just kind of means like a 3D shape. So these globular proteins, they're inserted into this lipid bilayer with the nonpolar segments in contact with the nonpolar interior. Because remember, like likes like. You learned that probably in chemistry. Nonpolar interacts with nonpolar, polar interacts with polar. So with the nonpolar segments in contact with the nonpolar interior of the bilayer, and the polar portions protruding out from the membrane, which means touching the water part, which that's okay, proteins can float in this lipid bilayer. And so that's what allows it. And so you have the nonpolar attaching, with the nonpolar, the polar going near the water, so everything is fine. And so that's why it allows this to basically occur.
Can I go to the nurse?
What'd you say?
Can I go to the nurse?
Yeah, sorry. It's just my right ear is partially deaf, so sometimes it's hard. Alright, so as for the phospholipid bilayer, every cell membrane is composed of phospholipids in a bilayer if it's a eukaryote. If it's going to end up being archaea, remember you can end up having a monolayer, which is different, and then bacteria can have a multitude of weird things. So when we discuss the bilayer, it's mainly discussing specifically eukaryotes. So bacteria and archaea bacteria are like a little stranger when it comes to this. So just know anything we're discussing right now is more referring to your plants, animals, fungi, proteins, like those 4 kingdoms within eukaryotes. So the phospholipid bilayer is going to have 3 main functions. So just make sure you know all 3. The first one is simply that the components themselves are going to allow flexibility. And so because of what they're made of, fats are very, very flexible. If you go and you took some adipose fat tissue out of you, it looks like a little blob and it moves around, and again, it's pretty gross, but compared to your muscle, it's very different. I don't advise going and watching a liposuction, but if you want to watch one, you will see what I mean. So the fat content in your body that they take out is very different from the muscle fibers. And so it has a lot of fluidity to it, and it just reminds me of a bowl of Jell-O, basically, that you're looking at. Your muscles would, again, you eat meat. That is a muscle you're eating. So when you're eating chicken or you're eating pork or you're eating turkey, that's a muscle. And so you know the toughness it has to it. And so again, when you're going and looking at a pile of Jell-O, that's very different. So again, these components allow a lot of the flexible matrix to be there. The other thing it's going to be able to do is it's imposing a barrier to not allow a lot of things in. So if the outside is polar, that means it's going to be really hard for non-polar things to get through, in other words, because they do not interact with one another. Again, there's some exceptions that we'll discuss, but for the most part, it does impose some bit of a barrier. And especially, do you really want a ton of stuff entering your cell and then having it explode? That would obviously be bad. And so it does prevent a lot of things from going and just getting inside. The other thing and last is it is permeable at least to some things, so some type of components, and we will go through those when we get to some of the next slides, in a few at least. And so certain types of gases and other things can pass through in and out, so it's not that it's a barrier and like nothing can get in or out. That wouldn't make any sense, but it does have the ability to at least allow permeability for certain things to get through. All right, so for the transmembrane proteins that are going to be there, proteins can float or they're embedded in the membrane. Like I told you, they can do either or. So if they're going to go and be attached to the cytoskeleton, that's going to be especially ones of intermediate filaments, they aren't budging. So they're going to make sure that they're definitely connected in some way, shape, or form. The ones that are connected to actin filaments, they're a lot more movable because remember, actin allows pinching and crawling and contractions and all this other stuff. So they can, move around a lot more. The ones connected to intermediate filaments, those aren't budging. So intermediate filaments is like a crazy thick rope, so you're not really going to go anywhere. So for these, the main purpose of them, and again, that's just like a category we use, this word transmembrane. It just means it goes from one side of the membrane to the other. And so this provides a transport of substances and information to cross the membrane. You will learn all about the different ones today. Receptors, electroreceptors, different types of channels for ions, there's like all these different things. But again, their main purpose is to allow certain substances in and out, and then also to allow certain information in and out. And so it just depends, because remember, electrical signals are technically information, different types of stimuli, things like that. And so you can definitely have these things going in and out of the cell. So the next one is the interior protein network. So this is going to refer to membranes that are structurally supported by an intracellular protein that reinforces the membrane shape. There's going to be like very specific ones that we'll be discussing. Spectrins is the one, but again, just to tell you again, so for the interior protein network, again, membranes are structurally supported by intracellular proteins. And remember, intracellular means inside a cell, intercellular means like cell-to-cell interactions. So intra, I-N-T-R-A, means inside the cell. And so those interactions are going to be taking place within the interior protein network. And so one of the ones that's going to affect the shape of blood cells is spectrin, which is written there. That's what gives red blood cells their concave shape. So a lot of cells will look like this and are going to be circular, but if you look at a red blood cell, it actually has a concave shape to it. And so this concavity that's there is due to the anterior protein network containing a protein called spectrins. And so it can actually change the shape that's going to be present. Again, there's all different types. The examples, they give you a few on a chart, but there are technically even more that are going to be existing. So the next one is going to be cell surface markers. So for this one, remember I told you that you need the equivalent of a flag on your cell to say, hey, you know, this is something that is foreign, I need to be destroyed, or hey, this cell is mine, self-recognition, don't destroy me. And so there's going to be glycoproteins and glycolipids that could be these markers. So remember, that word glyco and protein means some carbohydrate attached to a protein. Glycolipids are some type of carbohydrate attached to a fat. So we've already discussed that in other chapters, but those are just going to be markers and telling you what your cell is. So in other words, for cell surface markers, they're going to be going and telling it identity markers, like what type of cell is this? Is this a cell that belongs in your body? Should this be flagged for destruction? Is this going to be an old type of cell that should be destroyed? And so it will tell it all the information it needs by going and putting a tag on it. So kind of like if somebody wears name tags, you can know their name and find out who they are. And so it's that general premise of what's taking place here. So this is the basic breakdown of all the four major components. It's much more complex than this, but this is just a general idea. Yeah.
Can you go over the flexo matrix and the phospholipid bilayer?
So for the phospholipid bilayer, you're going to end up having permeability to certain types of compounds, which we'll get to. You're going to end up having it be a flexible matrix of some type, and then it can be a barrier to a majority of things. So those would be the three. And then which was the other one you needed?
Just the description of the flexible matrix.
Oh, just simply that you can, in other words, have the ability, because you're not always having every membrane be attached to a cytoskeleton, it can actually move around. So just like that boats of a pond idea. So it's just, if it's connected to actin or something like that, even microtubules, you can have the things move around. It's only if it's connected to intermediate filaments, it can't budge. So, but for the most part, it can be very flexible in its shape. Yep.
Can you repeat what a fluid mosaic model is?
Oh, it's just all this.
Oh, it's not.
Yeah, so in other words, it's going to be everything that allows the movement of the proteins and everything else, like boats on a pond example. Yep.
Can you repeat what the membranes are supported by?
Which, like inside of here?
The anterior protein network.
Yeah, it's going to end up being things like spectrin was one example, but there's others. So it's going to be different types of proteins, and those proteins decide the shape of the cell itself. I think the example in your sheet, the chart only gives you two. I'm only going to cover spectrins though, just that one, because the others don't matter as much to me. But the interior protein network will literally decide the shape of the actual cell. And so depending upon what proteins are in there, it can actually change the shape that's going to end up being present. So for this one, these are more of the recent findings, which we call them recent findings, but I mean at this point it's been around for 10 years or so. So the plasma membrane is not going to be what we call homogeneous. It's not all the same. It's not like protein, phospholipid, protein, phospholipid, protein, phospholipid. Like it's not always going to be the same. There's a lot of stuff that's packed in there. There's a ton of glycolipids, glycoproteins, cholesterol, and a bunch of other stuff as well. So we just say it's not homogeneous. So it's going to end up being not just the same thing in a certain pattern all over the place for the outside. So it's going to contain something called microdomains. That's what these lipid rafts are. And so they're going to have very distinct lipid and protein compositions inside of them. So these lipid rafts are something newer that was discovered. And so again, we call these things microdomains. In other words, a very small area on the membrane that has a gigantic amount of glycolipids, glycoproteins, transmembrane proteins, cholesterol, and a ton of other stuff. And so they're just jam-packed. Yep.
So lipid rafts are just like a jam pack of like different proteins and lipids.
I'm actually going to give you the exact definition in a
Okay.
second. Yeah. I just wanted you to at least get like the general idea at first. But in other words, it's just not going to be the same thing, and it's not going to be like a lipid protein. Like it's going to be varied. So Lipid rafts are going to be heavily enriched in cholesterol and saturated fatty acids. They're usually going to have both, which remember, that means there could definitely be a lot of flexibility, but at the same time, a lot of rigidity depending upon the temp. So heavily enriched in cholesterol and saturated fatty acids, which make it thicker than the other areas of the plasma membrane, and even on an actual microscope, the farther you go, closer you go in magnification, you can actually see these things. And so again, it's going to be heavily enriched in these areas with a lot of cholesterol, a lot of saturated fatty acids, and it makes it literally look thicker than the rest of the plasma membrane, just like they're trying to show you in this picture. And so when they first started studying these things, they were like, I have no idea what this is. Like, why is it thicker in these areas? So after studying, they found that first off, they are fantastic for signal reception. Kind of like if one person is just waving one flag and wants to be rescued off an island, you know, they might be not exactly being found. It's just one tiny dot. But if you have 100 people waving flags and, you know, lighting bonfires and everything else, you're going to get rescued more likely by a plane because they'll find you. So that's why these lipid rafts are there. If you need better signal reception, you need a lot of lipids and proteins and a lot of your cell receptors basically present. And so that's one of the main things that this is used for, is cell reception. The second thing is apparently cell movement is much easier within these areas, I guess because, again, of the flexibility within those spots, but it's definitely used within cell movement of all different types of cells. And the reason they started studying these is because of HIV. So if you go and you have these lipid rafts, and they have a lot of these glycoproteins and glycolipids and ID markers, HIV can kind of go and target these spots, because again, if you are waving one flag, it is much harder to find you. If you're waving 100, it's easier. So in a sense, this is waving a giant flag of what type of cell it is. So if you are an HIV and you're looking to infect a macrophage or something else, it gives you like an open spot, and you're like, oh look, this is the one I'm supposed to go to. And so if you can block that lipid raft from going and being found, or if you can put like some type of, you know, ability to not let it connect, no more HIV ever, which means no more AIDS. So that's what they're currently working on, and there are medications that do this currently, and so that's why normally, even if someone is found to have HIV, the likelihood of them getting AIDS is almost zero as long as they stay on the medication. They might have to change the medication over time because the virus changes, but it's crazy expensive too. Yeah.
So lipid rafts are a liability to the body?
Yes. At the same time, they're good for us, because they can ID what type of cell we are, so your immune system normally will not attack yourself, because you need a very large flag that says what you are. But at the same time, you're telling your enemy what you are. So it's kind of like your enemy uses it against you.
Okay.
So that's what ends up happening. Autoimmune disorders, what they do is they actually target a lot of these areas, because your body is saying, this is what I am, but autoimmune disorders are like, I don't recognize this, and then it actually starts attacking it. And that's what like psoriasis is, ulcerative colitis, Crohn's disease, anything like that. So any of those things would be targeted and done in that manner. And so very similar with a lot of viruses, this can happen too. If it affects anything involving these areas, it's like really hard to take care of. But that's where the study of these things started to become about. Especially like, you know, nowadays, much more than before, was all having to do with studying HIV, kind of how it works and the entire process of it. So, membrane proteins, again, they have a lot of different types of interactions with the environment. So we're just gonna go down all the different types of transmembrane proteins. Lucky for you, a lot of the names of these things actually tell you what they do. So when you get this on a test, normally nobody has a problem with this as long as you just remember the key words. But if something's called a transporter, you should have a general idea. It's transporting something back and forth in and out of the cell. And so this is where the protein can be very selective on what allows certain substances to enter or leave a cell. You sometimes have channels that like only let one type of ion through, like just potassium, or you're gonna have just calcium. Like they can be very specific. They can work off electrical signals. They can have like all kinds of variations to them. But again, transporters are areas where the protein's selective, and it only allows certain substances to enter or to leave a cell in some way, shape, or form. And so again, these transporters are going to be the area where the protein's selective and only allows certain substances to enter or to go and leave a cell. So enzymes are the one you're doing a whole chapter on for chapter 6. these are easy too. They're just gonna be the thing that carries out any chemical reaction on the interior surface of the plasma membrane. Usually there's some kind of signal to tell it to start, or energy or something like that. But in general, they're the ones that carry out the chemical reaction. So if you want to do something, you need enzymes to do that majority of the time. Sometimes they need energy to be kickstarted, and sometimes they don't. Cell surface receptors, again, also easy. They are going to be the membranes that detect chemical messages with receptor proteins. And so any type of chemical message will basically attach, and then it will send a signal to your nucleus to do something. And so cell surface receptors, again, are going to be membranes which detect chemical messages, and they're going to basically go and send out a signal to your nucleus. Remember, all your DNA is in your nucleus. And so it can go and just say, do this, either an internal response, external response, but it has to do something. And it does it rather fast. So cell surface identity markers, the word tells you the definition. So these are markers which identify the cells to each other and tell your body what they are. They're ID tags. So they just tell you what you're actually looking at. And so what type of cell you are, that flag you're waving type of thing. And so a lot of your glycoproteins and lipids are in this category. And so again, there are markers which identify them to other cells. They're ID tags. It just tells you what's there. The other one, the cell adhesion proteins, so these specific proteins, they glue themselves to one another. It can be permanent or it can be temporary. So that's what it looks like when you're doing this type of thing. Remember, we discussed tight junctions, septate junctions, all that type of thing. That's like that general category of where that comes from, especially tight junctions. So you have one cell here, one cell here. If you want to connect the two cells to line each other, you need to connect them with something. And so when you do that with tight junctions, those are proteins. And so that's how you're connecting them and making sure they stay still. So again, cell adhesion proteins, all they're doing is just making sure that they're going to be very specific proteins that allow them to glue to each other. It can be permanent or it can be temporary. It just depends on what has to be done. And so sometimes you're attaching because it's your immune system trying to attach and, basically take care of you, but it can also just go and be something that's not going to be permanent. The last one is you use a lot of proteins to attach to the cytoskeleton, because remember, how would your membrane stay in the circle or in the shape it's in if you don't have it attached to something? So it anchors to the cytoskeleton, and remember, there's 3 different types of cytoskeleton. It can attach to actin filament, which it did in the picture. That allows a lot of movement. It can attach to the intermediate filaments, where it's staying right still, it's not moving or budging from that spot. And then it can move to microtubules, which normally it offloads some stuff, and then it can travel to wherever it has to travel. And so it allows the movement of different particles and stuff to occur, kind of usually like a railroad station. So you would have a microtubule attached, you basically have whatever you have, and you send it off on there. But again, any attachments to the cytoskeleton, it's just there to help the rigidity, to make sure the shape is whatever it's supposed to be. So different types of proteins make different types of shapes of those cell membranes. And so again, it just depends on what you're using, and it depends on what type of cytoskeleton you have too. So if you're going to have active filaments, intermediate filaments, or if you're going to end up having microtubules, it creates all different things. So this is where they start going into passive and active transport, diffusion and osmosis. This is going to end up being all different types of the proteins that kind of run this process. And we'll be getting to all of these on here and all the different types. But again, if you want to know how it works in the examples that we'll be discussing, it's like your cheat sheet again for studying. It's all on there. So diffusion, that you might have done already in grade school, and that's when you're just moving some type of particle from a high to low concentration, and it has nothing to do with the water itself that it might be sitting in, but it has to do with the actual concentration of the solutes, the particles of the physical thing you're looking at. And so again, if you are moving things from a high to low concentration, that's going to be diffusion. If you're technically going to not use energy, it's passive diffusion. Technically, you can send something against the concentration gradient, and that's active, and we'll get to it in a few slides. But again, the net movement requiring no energy is going to be from high to low concentration. That's known as simple diffusion, and that's going to be one that, again, requires no energy to kickstart this process. So what we say is it's going with the concentration gradient. So If it's going from a high to low concentration, it's going with the gradient because it wants to go in that direction. Active is the opposite, where you're going against a concentration gradient because you're going and putting it from low to high. Nothing wants to go from low to high concentration. It will not do that naturally. That's when you need energy. So simple diffusion is just from a high to low concentration. So again, if you're going to end up having a total of 20 particles over here, and you have 5 over here, you're going to slowly have these particles move in this direction until eventually it's going to be 10 and 10, and it's going to balance out. It doesn't mean that it just stops. It's just that we say it reaches equilibrium, and it might move around still, but this will be the general concentration that's going to result. And so, not 10 and 10, sorry. So I guess we're saying 12.5, 12.5. I guess we're taking half a particle. But you get the general idea. So I probably just should have put an even number. This tells you I'm tired. So that's what they're trying to show you over here is if you take a dropper, you put it in water, a very high concentration will be the very beginning of this, but if you let time allow, eventually you're going to have the particles space out all over the place. Are they going to be equally spaced? No, it doesn't work that way, but they will be enough that it's going to end up going and dissipating. And I probably have to use something more colorful too because it's hard to see. But again, when you're going to go and do this, it's from high to low concentration with the concentration gradient, and it's just the solids you're moving. It has nothing to do with water. And so it's just, that's like the medium that happens to be there in this case, but that doesn't matter. So what we say is the membrane transport is going to be very selective with this process because it depends on if you're using proteins to help with this process. If you're not using proteins, and I'll let that thing go actually, because I don't think compete with it. So just give me one second.
Hang up on it.
I can't hang up on
I
it?
think so. I think Zerber does that.
Oh my god, it's awesome.
Wait, what was
Just made my day.
concentration?
Huh?
Low to high concentration?
Yeah, it goes from a low to high concentration would be against a concentration gradient, and that's going to be active diffusion.
That
So,
takes energy.
low to
CAS
high
takes a
is
ton of
active.
energy.
So low to high is active?
It's active and it's against a concentration gradient. Definitely use that word against, because that will help a lot. But again, the net movement always occurs from areas of high to low concentration, same as simple diffusion equals a concentration gradient. So as we're going into how it's selective, there's going to be different membranes that kind of run this process sometimes. Some things really can just travel in and out of the membrane based on this, but not a lot. So for the most part, you do need them to go through proteins, those transmembrane proteins, different types, and that's what we're going to get to. But you normally do need energy to do this. So again, when something is called selectively permeable, it just means that your membrane doesn't just take everything in and out. There's going to be very selective channels that run this process. And so again, you're going to end up having carriers and channels that we will discuss. Some are even going to be controlled by energy and things like that if it's going in the opposite direction, but that's what you're going to use. So there's many, many, many different types. So the channel quantities, all I put on there basically is just trying to tell you that it's not just one type of transmembrane protein, there's actually a ton. We're only going into a few of them and that's it, but there's more than I'm even going to be stating. The idea of what the fusion is, and again, to watch the Amoeba Sisters actually really helps with this, to see the visual. That's what the video is on here, and I always use it for the study guide questions, because it actually ends up helping the test. Grades are usually pretty high on this one. So these would just be some examples, bless you, of some of the transmembrane proteins. And so if you're going to end up going and having to diffuse ions, there's ion channels. So again, the name pretty much tells you what it does. And so these are going to be when you want to move different types of ions in and out of the membrane itself. So you can have ones that are going to go and have cation ions move. Remember, cation ions mean positive ones. Anions mean negative, so sometimes excess electrons that are negatively charged can move in and out. If you're dealing with protons, that are going to be your cation ions. So like calcium is a cation ion, so you would need a cation ion channel, a Ca, +2. If you're going to be moving chlorine, chlorine's a -1, so you would need a chlorine, that would be an anion channel. So you just have to know the charge on it, and that tells you what type you'd be using. So normally, both of these are going to be repelled by the nonpolar section of the phospholipid bilayer, and that's why you need ion channels. So normally what you have is an inability to send a lot of these elements in and out of the cell very easily, because again, it is very selectively permeable due to charges. So you have these ion channels that allow the charged ions, the charged anions to go in and out. And so propulsion cannot occur if you have these ion channels, because normally that is what's happening because of the charges that are actually going to be on the membrane itself. And so ion channels allow it to move, yeah.
It can't occur because of what?
When you end up going and having charges, so you're gonna be repelled by the non-polar section of the phospholipid bilayer because it doesn't like charges. Because ionic bonds and all that stuff is always based on charges. Non-polar stuff hates anything with charges. So even if it's a plus or a minus, it's just like, it literally bounces off of it, is the problem. Yep.
What's that little like ball thing below the channel?
Oh, it's like a plug, literally. You know how like in your bathtub, you literally put a plug, like, that's literally, they put a plug. So, just to make sure. I don't know why it's on some string and it looks stupid, but yeah. So, that's like their idea of putting a plug in. So, normally it's just gonna be something that if you have an excess and you need to move it, it will naturally happen. You do not need any energy to kickstart this, and it just moves the ions from a high to low concentration. So some of the stuff in your body for making ATP actually uses exactly this. They use ion channels. So the ion channels themselves, like the actual definitions of these things, they're going to possess a hydrated interior. Remember, hydrated means water, because things that are charged love water because water is polar. So again, ion channels possess a hydrated interior. It spans the entire membrane inside of it, and it does not touch the hydrophobic tails of the phospholipid bilayer. So this way, everything that goes through it is protected. So this is hydrated inside of here. Remember, anything water-based is very friendly to charges, and so it can go and get inside of here if it's a high concentration and it wants to go to a low concentration. And so this will allow the movement to eventually have equilibrium, and that's when it inactivates. And so it's just going to simply work when you have to move these things from a high to low concentration. But again, it does have a hydrated interior, and that protects it from all of the actual things that can naturally go and attack it for the most part. So the determination, yep.
Wait, what does the dove touch?
It doesn't touch the hydrophobic tails. Because the hydrophobic tails that technically are next to that ion channel, that does not like charges. And you do not need those things trying to like bounce around inside your actual membrane and causing some problems. So when it comes to the determination of the movement of these things, there's going to be two things that decide this. So number one, the relative concentration on either side of the membrane will decide, are they coming in or are they going out? Like on this example, on the outside of the cell, there's a very high concentration of ions. So the natural, with the concentration gradient, it wants to go from high to low. So that's what triggers this thing to open. And again, there'll be certain stimuli that you'll learn eventually that why this happens with all different types of things called ligand and stuff. But for right now, it allows this movement to get the equilibrium. But that's only one factor. The other one is there's going to be a voltage on your membrane. You're made of electricity. I mean, you're an electric being. Why do you think an electric charge can also kill you if it's too high? Why do you think it can revive you when you're, going through shock or anything like that? And so this is going to allow you to come back because you need to charge your heart. It's electric. And so this charge that's naturally on here can actually make this thing open and close. And again, it can basically respond to the concentration that's there. So the charge that's naturally on the membrane and then the concentration of like where these actual ions are located, that decides if this thing is opening or closing. And so those two things you have to account for. If you're not accounting for both, then this is not happening. And so again, ion diffusions, I didn't forget that part. So ion diffusion is going to be very important because it has to do with a lot of the signaling that will take place in your body. So if you want to have a nervous system, you can't have one without having all different types of voltage gated channels. So your brain literally works on this. So this is how you send a lot of the signals within your own brain, especially with like calcium. Your muscles are going to work this way too. So again, it's going to be mainly though focused on your nervous system. You do not have a nervous system without these ion channels that can send things back and forth.
Sorry, but can you repeat the first thing that determines the direction of the net movement? It was voltage and what else?
The concentration gradient, or in other words, the concentration of where the solvents are.
Thank you.
Yeah. So if they're high inside the cell, they'll go out. If they're high outside the cell, they go in. So it just depends on which way. So you're just looking to where the solutes are themselves. So facilitated diffusion is actually pretty easy. All it means is that you have to use some type of protein to get stuff in and out. So this is going to be diffusion that's still going from a high to low concentration, uses absolutely no energy, but you have to be going and putting stuff through some type of transmembrane protein. So that's why if you compare this, all three of these are passive transport. That means none of this uses energy. They all go from a high to low concentration. The only difference between simple diffusion and facilitated diffusion is they are going to go and use ion channels and then other types of different proteins that can get stuff in and out. So these are going to be carriers, and we'll discuss them in a second. Simple diffusion though, high to low, it just goes naturally through, nothing here, and I'll discuss some of the examples of what that, what those are, what types. But if it uses a protein, it has to be called facilitated, because you're using a protein to facilitate the interaction. And so again, facilitated diffusion is going to be the net movement that always occurs from high to low concentration, same as every other passive transport, uses absolutely no energy, same as other transports. The only difference between simple diffusion and facilitated is you're using carriers and channels. That's it. So we already went over what the channels were, but as for the carriers, these are going to be any type of membrane protein which is going to transport anything else that is not going to be those charged particles. And so if you want to move sugars, if you want to move lipids, if you want to move anything else, basically amino acids, across the plasma membrane, you have to do it through a carrier because it can't fit through your plasma membrane. Your plasma membrane cannot allow sugars to go through. It needs a carrier to do it because it needs a bigger hole, basically. You're not fitting it through otherwise. That's why when you did your experiment, we're trying to get starch particles to move. Starch is massive. It is not going to go and move anywhere, and so that's why it did not disintegrate. Instead, the iodine came to the starch because the iodine is smaller and it can naturally go through any type of membrane, structure. That's what happened with the dialysis tubing. So the carriers, they can physically bind to the ions or solutes trying to pass through. That's why they're called facilitated. So they're going to naturally just go and attach to that area. And so they're going to be able to, again, connect in some way, shape, or form that they can allow them to go from high to low concentration, no energy, has to be a protein, and so it's usually not some type of normal ions. This normally transports a lot of the ions, so something charged. This is going to be more of the physical things like your amino acids, your sugars, lipids, things like that that can move in or out. So this is what this is trying to say, and this is one of the questions I had even asked in lab, and it is a little complex to answer, so I'm just going to try my best with this. All right, so when you go and compare facilitated diffusion and simple diffusion, the higher the concentration gradient, the faster it's going to happen, because if you have a really high concentration here and then a really low concentration here, this is going to want to travel in this to reach equilibrium, and again, it's going to be a linear line, so the higher the concentration gradient, generally speaking, the faster the rate of the uptake that this is going to happen. Remember, if this was like the same concentration in both places, it just basically reaches equilibrium. We just write it this way because technically things still move a little, just very slowly. This would be extremely fast. This would be almost non-existent and not really moving. And so this would be the area that would be closer to that 0 mark. This would be the area that's closer to the top of that green mark. So again, the speed that happens for simple diffusion is all dependent on the fact that the concentration gradient that is occurring, if there's a very large difference between the two places, you will have a very fast rate. If not, then you're not really going to have much of anything happening. So the annoying one is the facilitated diffusion. Generally speaking, if you are going to do this, you need proteins. It depends on the number of transmembrane proteins available, your ion channels and your carriers. That's what decides the speed. The reason you see this, we call it like a logistic growth model or a carrying capacity. When you reach the maximum number of what you have for enzymes, this cannot go any faster. So same thing, if you want to go and take a trip and get people on a bus, let's say the bus fits 25 people and you have 25 people, but yet there's also another 25 that have to go on the trip. Those 25 people that are left extra are not going until they basically drop off the first bunch, come all the way back and drop off the second bunch. So if you were just able to have cars instead and everyone drove themselves, you get there like this. But by taking the buses and only fitting a certain amount of people, you are limited on the amount you can take at one time. That's what you're limited by here, is the number of proteins. So the number of carriers, the number of channels. So in the beginning, when you have a lot of these carriers and channels available, you can quickly go and basically begin diffusion. But the second you start filling them all up, it doesn't matter if the concentration gradient is high as can be, you maybe only have like 5 carriers and channels. So it can only fit in 5 things at a time. It doesn't matter if there's 100 on one side and 3 on the other. It doesn't matter that the concentration gradient is so high. It's not going to make it any quicker. So simple diffusion, the speed of it is just decided by the concentration gradient that's there, and the amount that you have of the solutes on each side, that decides the speed. But not facilitated diffusion, you are simply limited by the number of carriers and channels you have for that specific interaction. And so the speed in the beginning can be quite fast because they fly on through when they go through those carriers and channels much faster than they can do through the actual bilayer. But the only problem with that is you are limited to the number of spaces. And so it can actually end up taking longer if, again, you are using up every single carrier and channel and you can only do so many at once. So, the main characteristics I wrote there, the reason I wrote this in that one position is simply that I want you to know for facilitated diffusion what the 3 main characteristics are for that one. So first off, it is specific. So that means that a given carrier transports only certain molecules and ions. If there's something is called a calcium, ion channel, guess what? It's only moving calcium. So if something is going to move glucose, it's only moving glucose. It's very specific. So a given carrier transports only certain molecules and that's it. So it's specific to just a certain type of molecule or ion or something like that. It is passive is the easiest one. So the direction of the movement is determined by the concentration gradient. And so you are not using energy. So if it is called facilitated diffusion, it means it's passive because it's not active transport. No energy is being needed. You are simply moving something from high to low concentration. But again, the speed at which it happens is all dependent on the number of carriers and channels. So lastly, that thing that you see, what we call logistic growth models, carrying capacity, all it means is it saturates. In other words, you only have so many carriers and channels. You can only do so many of these reactions at a time. It saturates the system. It cannot go any faster after that point. So in other words, if all relevant protein carriers are in use, it increases the concentration gradient. Like if you increase, the concentration gradient, you still can't go faster because you're only using so many places. Why do you think New York City has such traffic? We have 1 million cars that want to get through, but we only have the Holland Tunnel, Tappan Zee, if you want to go in the one area though, I think it's called Mary Cuomo now. You have the, George Washington, and you have the Lincoln Tunnel. That's it. So you have 4 entry points for millions of people. And so that's why congestion happens, because you only have so many entry points. I don't think you're about to swim across the Hudson. And so again, that means it's saturating the system. You only have so many carriers and channels, and so you can only go so fast. And again, it's not a constant rate like the simple diffusion. And so that's what it means. If all relevant proteins are in use, increasing the concentration gradient does absolutely nothing. And so again, if you ended up getting this picture on the chart, just kind of know what it means and why those two are different. So that rate of diffusion will vary, but this one is based on the carriers and channels eventually. This one is based on just the concentration gradient alone. So technically one can be faster at the beginning, but that's because again, your lipid bilayer is not the easiest thing to get through. So that's why in the beginning, this could take a little bit of effort if the concentration gradient isn't very high. But here, it can go very quickly because it's a giant opening, there's a giant hole through those ion channels. So osmosis is going to end up going and being the next one. And so this is the water one. This is the one that we discussed. You just did a lab on this one as well. And so this is the one, I have this in one more slide after I want to get through today. So, this video I would highly recommend as well. This is all about osmosis, how it works, what it looks like when you're trying to go and, have this process happen. But please don't confuse the definition for this for diffusion. So osmosis is the movement of water towards a higher concentration. So in other words, if you saw these two concentrations, the water, the H2O, would actually be moving in this direction, because the whole point is to dilute it in some way, shape, or form. Here, the water would just go again and stay pretty much the same. It wouldn't really be doing anything. But it's the movement of the water itself. It has nothing to do with the movement of ions any way, shape, or form. It's the net water movement toward a higher solute concentration by a diffusion process, but it's the water itself. So you have to look to where the solutes are. That tells you where the water goes. That's why, again, in your lab, if you had something that was going to be technically a very high concentration of, solutes in here, low out here, if you did this to a red blood cell, the water went in, eventually tons of water come in, and the cell is going to go and explode, and you're going to have like giant openings, and it's going to be bad. And so this would lead to lysis. It would lead to an explosion of a cell if it's going to end up being something that is going to be a animal cell. Because remember, plant cells can't explode. And so this would be, again, the water movement that causes this to happen and this explosion process. So, water is facilitated in and out by aquaporins. That's what those things are. They're very special channels for water. The name tells you, it means water pores, water holes. And so aquaporins are special channels for water. This facilitates water movement very easily. So, the concentration of all solutes in a solution, that determines what's called the osmotic concentration, how you're moving this water. So again, the concentration of all solutes in a solution determines the osmotic concentration of the solution. So again, I'll just keep on saying, just make sure you have it. So the concentration of all solutes in a solution, that determines the osmotic concentration. So again, the concentration of all solutes in a solution determine the osmotic concentration of the solution. All right, so we've already gone over this, so you should have these definitions. Your lab was just handed back to you, so you should be fine. So I'm not going to spend a million years on this. But just remember, hyperosmotic means the solution with the higher concentration. Anything that's going to be called hypoosmotic has the lower concentration, technically, of the solutes. Osmotic means that it would be equal with both. So this would be osmotic. This would have been something that was going to be technically hypertonic. This would be hypotonic. So those words will always be the same thing. You can even call it hyperosmotic, hypertonic. It doesn't matter what the stupid second word is. That first word is what matters. So hyper is always going to be the one with the higher solute concentration. Hypo always means the one with the lower solute concentration. And iso always is going to be the equal one. So again, those definitions you actually just got handed back, so I just didn't want to spend a lot of time on those, because I would like to at least watch one of the extra credit, and we really only have the one slide left, and that's it. So this is not that bad. All right. So for this one, you're going to have something in your cells called osmotic pressure. This is the pressure that must be applied to stop the osmotic movement of water across a membrane. In other words, reach equilibrium. So, the osmotic pressure, this is going to be the pressure that must be applied to stop the osmotic movement of water. Oh, my bag's filthy. All right, so again, the osmotic pressure is the pressure that must be applied to stop the osmotic movement of water across a membrane and not allow it to continue. So again, the pressure that must be applied to stop the osmotic movement of water across a membrane, that's going to be osmotic pressure. So, there's two other pressures here. I know I'm jumping in between, but it's easier for me to give you the definitions and then describe these things. So the hydrostatic pressure, this is the pressure pushing against the cytoplasm of the cell membrane. So again, if you have a lot of water coming in, this means that this would have very high hydrostatic pressure. It's pushing against the cell membrane itself. So again, it's going to end up causing this to happen. If you had the reverse occurring, so instead, if you were going to end up having a high solute concentration here and a small amount here, and water leaves the cell and eventually it shrinks and it crenates, that that means we have low hydrostatic pressure because this is not being pushed, so it would not happen. And so again, low hydrostatic is on the left, high hydrostatic is on the right, and all hydrostatic pressure means is the pressure of the cytoplasm pushing against the cell membrane. And so it can basically tell you, if you said, hey, if I have a high hydrostatic pressure, what type of state would a cell membrane be in if it was an animal cell? And you would say, look, there's a chance it will do lysis because it's going to be very high here. And so we do not want that. We want iso, technically, is what we like. So I'm going down to the turgor pressure one because I'd rather you know those two first before we go through these. So turgor pressure, first off, is only in anything that has a cell wall. So plants, we don't have cell walls. Some protists also have them, and fungus also have something very similar. But in general, turgor pressure can only apply to something with a cell wall, and that's it. So plants that have a high amount of solids within their cells, okay, you have water come in, and remember, they have high hydrostatic pressure if this was happening, but they're also going to end up having a cell wall. So you have a membrane that's going to be on the inside, so if you have water coming in and it's pushing out on all the corners, we would say high hydrostatic pressure. But if it's also pushing out and it's going to be very turgid, very filled with water, we say it's going to be very high turgor pressure. It means there's a high amount of pressure pushing against the cell wall. That's why you can't have that because you don't have a cell wall. So turgor pressure is only having to do with the word cell wall. It is the water pushing against that, from vacuoles or from cytoplasms or whatever's in there, pushing against the cell wall. So the hydrostatic pressure is the pressure against the cell membrane, and then the turgor pressure is going to be the pressure against the cell wall. And so again, plant cells can handle things differently than we do. So,
So like, the more the turgor pressure is, the better for like plants.
100%, and that's what we're going to go through right now. So, maintaining the osmotic balance, I will get to in a minute. I actually want to tell you the difference between these first. So, this is what the video and this is what the lab was trying to explain to you. If you have a lot of water coming inside a cell, we use the term lysed for animal cells because they can explode. They can't explode if they're going to be a plant cell. So lysed just simply means that the cell is going to burst. Heavy lysis means it's about to burst, but either way, it's bursting in some way, shape, or form. So if you have a lot of water coming in, then it's going to be bad. So if you are hypertonic in here, and you're going to be hypotonic on the outside, this is not what red blood cells like. But plant cells love this because they have very high trigger pressure, very high hydrostatic pressure. They are in a turgid state, that's its favorite state, and it means simply that it is filled with water, its vacuole is filled inside of there, it's going to be fine. We can't handle that. So we cannot handle high hydrostatic pressure, we explode. Our cell membrane, is not the same thing as a cell wall. This is now the opposite. Flaccid simply means that it's not going to be, it might start to wilt a little bit, in other words. So plants actually don't like isotonic solutions. They only want to be turgid. So if you end up having the same amount of water on the inside and outside, it does not like that. We do. So this is our favorite one because, again, we do not want to explode and we don't want to shrink. So we're like in this, middle ground. For plants, this would be a wilting plant. It would not be dead yet, but it would start to wilt, so you'd want to make sure you water it. And so again, this is just when there's equal amounts of water on both sides. So shriveled, or what we also call crenate, is going to be the one that if you're having a red blood cell sitting in a hypertonic solution, and that's what we don't want to happen. And the chances of this coming back are not very good. Same thing for plant cells. Technically, if you add water, it might go back. But for this one, again, plasmolyzed means it's going to have a really low hydrostatic pressure, really low turgor pressure. You're losing all your water. That plant cell is going to be dead over time. Again, you even have the contraction of the cell membrane literally ripping off. You have the tonoplast of the vacuole being destroyed. It's just not good. So the term plasmolyzed, though, remember, is only for plant cells. Turgid is only for plant cells. Lysed is only for animal cells, and crenate, which means shriveled, is only for animal cells as well. Because I'll use those terms if you get a test and those questions are chosen. I make sure that certain terminology is used for the different things. So, lastly, if you want to go and maintain this osmotic balance, how do you even do this if you're tiny little organisms too? So there's this process called excrusion, that's what's written on there. So this is when, say you are a single-celled organism. So naturally, if you're a paramecium and you have like all this cilia, whatever else, you're just swimming around, you're just one cell, what they'll actually have is they're gonna have this like contractile vacuole, it's called. This contractile vacuole will actually actively pump out the water. Because remember, if you are an organism, there's normally going to be a higher concentration of solutes in there compared to here. So water wants to naturally travel in, but if too much water travels in, you go kablooey and you explode. So it can actively pump the water out if that is the problem, and that process is called excrusion. So contractile vacuoles, just to make sure you have the wording. So again, this is going to be what paramecium and some of the single-celled organisms have, are these things where they can actually take in the water and then they can actively pump it out at the same time to make sure through these like specialized pores that you're not going to end up going and having the thing explode. But again, this only happens if you end up having a hypertonic solution inside here and hypo on the outside. But it has this ability to do that. Then the other thing I put on there is isomonic solutions. Some organisms, when they're sitting in salt water, remember what would normally happen is the opposite. If you happen to be sitting in salt water, you are going to end up having, actually this will fall out, you will end up having a high concentration here, low here, which means water wants to actively leave. You don't want water to leave your cells because you're going to shrivel up. So what sharks do is they actually maintain an isoosmotic balance between the salt water. They actually intake some of the salt water into their own cells to make it match with the ocean. Not a lot of marine organisms can do this, but sharks can. And so again, what they do is when they live in the ocean, they adjust their internal concentration of solutes to match that of the surrounding seawater. Remember, that's what ISO means. ISO means to be the same between the two. So it can actively take in some of the salts just to make sure it maintains it. Normal fish do not do that. Bony fish don't. It's just sharks that have this ability to do this. And so again, the turgor pressure one I already went over, so you already have the information for that. Plant versus animals, we already went over for that, so you're fine with that too. It's just that those other two were just a little bit separate on how you maintain this balance. But again, for the most part here, if you just simply water the plant within like a day, it'd probably be fine. This, there's no coming back from it. When we lyse or we shrivel up, the red blood cells don't come back. So a lot of the time your proteins are already destroyed, so they're not really going to be able to, so they end up just dying and that's it. I'm trying to think. So it's 12:01 now. Uh-huh. Yeah. I'm trying to think of just which video, and let me look at the times on each of them. I would like to at least try to show you one of them if possible, so you can try to get some extra credit. So let me just see. So we have one that's 9 minutes, 8 minutes, and 10 minutes. All right, so if you had to choose, do you want to do the 8-minute because it's the shortest?
Is it easier?
They're all pretty easy. We have inside the cell membrane 9